Good morning, everyone. Let's get started with the class. Are there any questions before I switch to the lecture screen? So traditionally, I'll start with a few reminders. We have quiz 7. It is open and is due on this Friday. And next quiz opens on Saturday early morning, and it's going to be due in a Friday, next Friday, like the Friday after this one. And there is a participation assignment that is going to be due today. So don't forget to go to modules, click on the participation assignment and take an assignment. It's a short quiz this time. Jennifer, did you have a question? I see you're unmuted. Right. Okay. Moving on. So today we are going to move into another section of the course dedicated to the evolution and diversity. And today I'm going to cover evidence of evolution. You will see that in the schedule that tomorrow I'm scheduled to cover it as well, but we'll see how today goes and if I might cover everything today and move on to the next topic. So many types of clues enable us to hypothesize about how modern species evolved from the extinct ancestors and understand relationship among the organisms that live today. A lot of those clues come from the anatomy of the various species. So, and a lot of those clues are provided by fossils. And with the development of the molecular techniques, a lot of those clues are also provided by the DNA. So we can compare DNA of one species with another and evaluate how closely related they are. In fact, the phylogenetic trees, which you have seen several weeks ago, which we studied several weeks ago, are all developed based on the genetic similarity between species. We're able to infer some information about the common ancestors based on the similarity between various species DNA. So I'll start from the oldies. Okay, one of the sources for evidence of evolution is provided by the paleontology, which is the science that studies the past life. So one of the clues of evolution to evolution are fossils, and fossils technically are the remains of ancient organisms. Um, so how do fossils form? You might ask is usually the fossils are found in the areas where there used to be an ocean or a sea or a lake, or a bog even. So fossils form when a living organism dies and either is buried under the thick sediment in the ocean or gets to like into a bog. Um, the crucial thing for a fossil to form is the absence of oxygen. Okay, so whenever a tree dies, let's say, if it falls onto the ground, then it decomposes and eventually its wood is incorporated into the soil layer. So basically soil under our feet is the organic matter, and the soil is the decomposed organic matter from the living plants and and animals to some extent, but mostly plants. Now, for the fossils form when a tree, let's say, falls into a bog or into a lake, and it sinks to the bottom, and eventually it is buried under the sediment. So, but it's not decomposing. It decomposes to a certain extent, but not much, because in the absence of oxygen, there are very little microbes that can survive that would prey on that dead wood, on the dead organic matter. So the tree remains virtually intact if it falls into a lake and is buried under the sediment. But over time, the organic matter is substituted with silica, for instance, with another non-mineral compounds. And this process of substitution of the organic molecules with inorganic minerals is called fossilization. So the fossils can be divided into two categories. There are true fossils and just, well, untrue fossils. With the true fossils, there is no organic matter at all within a fossil, and with not so true fossil, there is some organic matter. So the main distinction between the two is that from the true fossil, you cannot really extract any of the ancient DNA, but from more recent fossils, you can find some remains of the DNA. All right, so I'm going to get to the picture here, and I'm going to explain what is actually shown in here. So this is the ancient fossilized plant, and it's called horsetail. This is the petrified wood. If you've been to the Washington State, there's big park in there by the Columbia River, closer to the western part of the Washington State, and there are literally these petrified pieces of wood laying around, and this is what they look like. So you can really in some of the specimen of the petrified wood, you can even see the tree rings. So you can kind of compare the width of the tree rings and infer how the trees grew many years ago. So you can even infer maybe like temperature or weather and so on. This is the fossilized dinosaur egg. Like if I were to zoom in, you would see a little bitty baby dinosaur in the egg. This is the fossilized poop from an ancient turtle. This is an arthropod, fossilized arthropod, which is called a trilobite. It's ancient, it's extinct now, but it used to be very widespread around our planet. Okay, this is a fossilized fish, as you can guess. This is a pretty straightforward one. This is fossilized leaf of ginkgo biloba. It's a plant. You can see live ginkgos around, and fun fact: the live ginkgos that you can see planted in parks, it's only male specimens. Like there are there are very little females, because the females produce the seeds that are really stinky. They stink up the neighborhood. Okay, so whenever the ginkgos are planted for beautification of the area, only male specimens are planted. And finally, probably if you visited any science museum, there are usually some sort of display of the dinosaur bones. So this is the actual dinosaur skeleton, fossilized dinosaur skeleton. And what's the name of the dinosaur? I'm blanking on it. Triceratops, that's the name. So are there any questions about how fossils form? Okay, no questions. So the fossil, let me see something in chat. Okay, Chantal is asking to redefine fossilization. Okay, so fossilization is the process of conversion of the organic matter to inorganic matter. So basically, the organic matter is washed out or is substituted with minerals like silica or calcite. And if you're wondering what silica is, the sand is silica, okay? And calcite are the minerals with calcium present in them. So during fossilization, the shape of whatever object is being fossilized is preserved. The inner structures are preserved, but instead of organic matter gets gradually substituted with inorganic matter, with minerals. And that is why fossils become very hard. There's basically, they feel like a rock. They don't feel like this turtle poop here, this fossilized turtle poop. It feels like a rock. It doesn't feel like poop, you know, it doesn't even crumble. It's just a solid kind of slab of fossilized poop. Same with other fossils. If you were to touch them, you wouldn't be able to guess that this was a fish. You wouldn't be able to guess that this was an actual leaf. You wouldn't be able that this thing was alive. And if you touched a fossilized tree, you wouldn't be able to tell that, oh, this was a tree, because it feels completely different. Like, to the touch, it feels like a stone, due to mineralizations, to substitution of organic matter with minerals. Chantel, did that clarify the fossilization for you? Great. So fossils record evolution. They provide a collection of ancient remains of plants, animals, and microbes, and they also allow researchers to test predictions about evolution. And here's another example of fossils, which are found on the bottom of, in former sediments of the former ocean. Okay, so these marine fossils are very common in the central United States, indicating that that area once covered by the ocean. Um, I wonder if you've covered in school, like, what else is can be commonly found in the areas which were formerly covered by the ocean. Like, apart from fossils in the central United States, what else can be found in the central United States that points at it being formerly covered by the ocean? Anyone? Christina says garbage. No, oil. Oil is the correct answer. So in the former in the areas formerly covered by the ocean, oil is very commonly found. And the reason for that is the oil is basically the ancient organic matter. It was converted from plants, basically. Practically all of the oil comes from the so-called Carboniferous period in Earth's history, where when Earth was covered with this lush vegetation. And whenever this lush vegetation died out, a lot of it ended up in the ocean. And if there was no access of oxygen to the vegetation, then it's under conditions of heat and pressure, it turned into oil, well, and coal. All right. So well, some of the fossils are called transition fossils, or classified as transition fossils, and these transition fossils provide some knowledge about the links that demonstrate common ancestry. So before the molecular technique came into play for studying evolution, so if you have access to the genetic material, it's pretty straightforward to kind of sequence the DNA and conduct the intercomparison of the DNA of various species to find out how related they are. But before the DNA study was what became popular, and it didn't become popular that long ago. I mean, DNA was discovered in the 50s. But before that, paleontologists relied solely on fossils to study evolution. And the fossils were often dated, and the various fossilized organisms were intercompared with each other, with anatomical, like specifically their anatomical features, because you couldn't do much more with the fossils. And the combination of dating the fossil and comparing their anatomy allowed to build some evolutionary trees, and kind of allowed to hypothesize how did life evolve on Earth. So this is an example of the transition fossil, and it's a fossil of a dinosaur called Protarchaeopteryx. And this fossil has a beak. It had feathers, which you can kind of see the fossilized feathers here around the wings, and the fossilized feathers around the what do you call it, legs, bird legs. And these are also traits of the modern birds. So this Protarchaeopteryx fossil is considered a transition species, a transition fossil between the dinosaurs and the birds. So it provides a clue that birds evolved from the dinosaurs. And uh, what else? Well, that's the main thing about to know about transition fossils. Basically, transition fossils are fossils that provide a link between one species and another. Like it provides a clue how the life evolved. They provide a clue how life evolved. So I mentioned dating. Dating is important in building the evolutionary trees. And there are two types of dating because we need to know we need to know what species came before which species came before which in order to build the evolutionary tree and go back to finding the common ancestors. Okay, um, so there are two types of dating. The first type is relative dating, and the second type is absolute dating. The relative dating, as the name suggests, is not precise. It estimates fossil age using rock layers. And by age, I use the age here is used in sort of an abstract way because you cannot identify the age, rather you can identify which species lived earlier than any given other species. So if you start digging a hole in the ground, the layers that are closer to the surface would be more recent layers, and the layers closer, like the deeper you go, the older the soil layers or the rock layers will be. So if a paleontologist goes out and starts digging a hole down, then the fossils that would be discovered closer to the Earth's surface would be more recent. And the deeper the paleontologist would go, the more ancient, the older species they would find. Okay. So does it make sense with this relative dating that the whatever you find that is closer to the Earth's surface will be more recent than what you find deeper in the ground? Any questions about that? So if you're wondering why that is, I'm gonna say I'm gonna introduce the concept of erosion here. So soil constantly erodes, and it erodes from one area and transfers into another area, uh, and we are constantly being kind of covered by the thin layer of sediment. Our Earth's surface is like in some areas is covered by a thin layer of sediment, and like on a yearly or decadal scale, the sediment is very insignificant, but over the scale of millions of years or like tens of thousands of years, this layer of the sediment becomes more significant. So, for instance, like in in, um, in some ancient cities, whenever buildings are built in some ancient cities, lots of times during the building process, the builders would find like remains of ancient civilization below. And this ancient civilization, basically, once it became extinct, it literally got covered by the sediment over the thousands of years. And but like there would be solid layer of soil on top or a solid layer of rock, but and this layer formed over like maybe 2,000 of years or 3,000 of years. So this should give you an idea that it doesn't take too long to build a substantial layer of sediment and kind of cover up the ancient city if like it existed in the past. So pretty exciting stuff. Like if you ever go to Europe or to European cities or Turkey has lots of remains of ancient civilizations. Go to take a tour in there. It's fascinating to see how deep these ancient cities were and like how um, apart from other quirks of the civilizations. And the second type of dating is called absolute dating. The absolute dating estimates fossil age using chemistry or properties of isotopes. So this picture here, um, basically it describes the decay of the radioactive isotope of carbon or carbon-14. Does anybody remember what does isotope mean? What does C14 means and how is it different from just C?
It's an isotope and it has additional neutrons.
That's exactly right. So the carbon atom usually has most of the carbon atoms on Earth. How many neutrons they have? That's exactly right, Dylan. So 12 is the most common number of neutrons, and 13 is going to be another variation, but 14 is the radioactive form of carbon. So because it's radioactive, it's unstable. Okay, over time, C14 converts into nitrogen 14. So you like, and this property, this decay, allows us to identify the age of something. If we analyze carbon content of some matter, and we basically analyze the C14 content of some object, we can identify how long ago did this formerly living organism died. So carbon 14 decays exponentially. Like while an organism is alive, its carbon 14 content remains constant. But after an organism dies, this carbon 14 content in this formerly living organisms becomes to decline. It declines gradually. So um, in this graph, it is the exponential decay is shown in terms of the half-lives, which I think is more user-friendly way to show it than just years. So the half-lives is the amount of time that it takes for the half of the isotope to be gone from the matter. So for carbon 14, the half-life of carbon 14 is 5,730 years. So after 5,730 years, there will be half of the carbon 14 content in the formerly living organism compared to the original content when the organism was alive. After 11,460 years, or two half-lives, there will be 25% of the carbon 14 left compared to the original content of the carbon 14. So after note that um, two half-lives will be like 50% divided by two. And whenever three half-lives are passed to identify how much carbon content, how much carbon 14 is contained in the formerly living organism, you would take 25 and divide it by 5. So it's 12.5%. Uh, so Christine is lost, so is Lauren. Okay, so imagine you have a living organism, and we're operating, the y-axis here is the relative content of the carbon 14 to the living organism. So you have you have a fish, okay? You have a fish that swims around and it has relative amount of carbon-14 of 100%. So while it's living, we take the content of the carbon-14 as 100%, which does not mean that all of the carbon in the fish is carbon-14. It's the y-axis is relative to carbon-14 content relative to the carbon-14 content in the live organism. So while the organism is alive, by default, relative content of carbon-14 is 100%. Once the organism dies, the amount of carbon-14 within this organism will start to decline. Okay, so the relative, did someone say something? No, okay. After 5,730 years, the relative carbon-14 content in this dead fish that has started to fossilize, I'm sure, is going to be 50% of what it originally was. After 2 half-lives, or 11,460 years, 25% of original C14 is going to remain in this fossilized fish. After 3 half-lives, which is 5,730 years multiplied by 3, 12.5% of the original carbon content will remain in this fossilized fish. So if we know the original content, by identifying the carbon-14 content in the fossil, we can infer how many years ago did it die, okay, or how many years ago it lived. So if we find a fossil and we find that it contains 12.5% of the 14C contained in live organisms, we can infer that it lived 5,730 years multiplied by 3. Okay, because 12.5% of the original carbon-14 content corresponds to 3 half-lives of carbon-14. Was that any clear? It's a complicated topic, but it's important to understand. Okay, I heard from Lauren. Who else? Who else is still confused about, or who is still confused about how we use carbon-14 properties, property of carbon-14 for dating?
Okay, so if we want to determine how old when the fossil died, we will be multiplying, for instance, 5,730 years by 12.5, 25, 50, or 100, or by 1, 2, 3, 4, the death year. I think you muted yourself. We can't hear you.
Sorry. So you would be multiplying 5,730 years by 1, 2, 3, or 4. Okay, maybe this figure is more confusing than the other one I had in mind. Anyway, so I'm gonna go through the example again, and Diamond, you tell me whether it was clear or not. So you dig up a fossilized fish, and you want to study how old it is, how long ago did it live. So because it's fossilized, you immediately have a clue. Okay, it didn't live like 1 or 2 years. It didn't die 1 or 2 years ago. It takes a long time for something to fossilize. So you take a sample and you analyze carbon-14 content in it. And you take a live fish, this is something I haven't mentioned before, and you take a live fish and you analyze the carbon-14 content in that fish. So, you know, your carbon-14 content of the live fish will be your reference, okay, will be your 100%. Now, the carbon-14 content in the fossilized fish will fall somewhere along that curve. So it can be 50% of the live fish content, it can be 25% of the live fish content, or it could be 12.5%, or somewhere along that curve, okay? So once you've identified where that carbon content in the fossilized fish falls on this curve, let's say it falls right here. So the carbon content in the fossilized fish is 25%. Then
Okay.
up
Would you
to,
be telling us what the carbon content? I'm sorry. Would you be telling us what the carbon content is, or how would we figure that part out? Like if it's
I
25%.
will formulate the question in a way that, you know, you shouldn't require any information apart from understanding how radioactive decay works and what would you need to do to identify um, the age. I
Okay.
will
I'm
tell you
sorry.
everything you need to know when I ask you questions about that. Okay, and just to finish up the sentence, if your carbon content in the fossilized fish is 25% of that of the live fish, um, it means that your fossilized fish lived 2 times 5,730 years ago, which is 11,460 years ago. Anybody's confused? Okay, so um, there is a little quirk that I would like to go over with this carbon dating, uh, which, which has to do with adjustments. So we consider the carbon-14 content in live organism is like the same as it was 12,000 years ago or 50,000 years ago and so on. This is technically not true. We need to adjust, like scientists need to adjust this assumption, like before they date the fossils. The assumption that the carbon-14 content in the living organism is the same as it was, 50 years ago, 50,000 years ago. And that is because in the early 20th century or mid 20th century, lots of bomb testing was going on, and this bomb testing basically produced lots of carbon-14. They produced a spike in atmospheric carbon-14 because of like all the explosions and all of the disturbance that it caused in the atomic structure. So of carbon. Uh, so scientists need to adjust for this spike in carbon-14 from 40s to 60s. But it's basically easily done, but it's something to keep in mind that carbon-14 in the present living organisms is not technically the same as it was in the organisms 50,000 years ago. So if you were to date something, you would first need to adjust your reference carbon-14 by a curve of by the spike in the 40s and 60s in carbon-14, as well as the decay curve since then. Okay, I see a question in chat. Can you also calculate how long they lived before death also? No, technically you cannot, because while an organism is alive, its carbon-14 content does not change. And the reason it does not change is because uh, the the organism constantly ingests food, and this food like contains this carbon-14. So over like while the organism is alive, it's constantly resupplies itself with fresh carbon-14. So we don't see the radioactive decay, like the decay of carbon-14 as the organism lives. We only see it after the organism stops eating. So we see it after the organism dies. So we cannot use carbon dating to figure out how long an organism lived. We only can figure out how long ago did it live. Any other questions about this? All right, so fossils are dated by measuring the isotope carbon-14. And so usually fossils that have lived less than 100,000 years ago are dated with carbon-14, that I should add, because of the relatively short half-life of carbon-14, we cannot date a fossil that lived, I don't know, a million years ago, because it's not going to be precise. The half life is relatively short for the fossil. So whenever we have a fossil that might have lived millions of years ago, we don't use carbon-14. We use other isotopes like PB. What is it called? The
I
lead.
was gonna ask, is there like a certain general number of half-lives that you get to where like like say like 7 half-lives into carbon-14, like it's no longer like as accurate as,
Yeah,
4 half-lives in,
right.
because the
So
curve
after
gets
you
so
get
small.
that's a
Okay.
very good point, Dylan. So you see the curve kind of becomes flatter and flatter the more half-lives you get. So after like 4 half-lives, it becomes increasingly less precise. The carbon dating method becomes increasingly less precise. So usually after 4 half-lives, you just switch to another dating method, not carbon dating, but something else like lead dating or strontium dating or something else. All right. So as I just mentioned a minute ago, as while the organism is alive, it eats and constantly replenishes its carbon-14 content. But after it dies, the carbon-14 starts decaying into nitrogen-14, and the carbon-14 content within a dead organism gradually declines. And this gradual decline allows us um, to kind of back engineer the age of the dead organism, or the length of time that this organism has remained dead. Okay, so this figure kind of illustrates what I've discussed before. Are there any questions about carbon dating and why carbon-14 remains constant while the organism is alive, and how, like, basically how we figure out the age since death, the time since death? Okay, moving on. Another thing that sheds some light on evolutionary events is biogeography. So this sometimes, in some places, the the diversity, the kind of species that are present in a place with the same temperature, with the same moisture, with the same food availability, and with this with the same basic, virtually the same conditions. Sometimes the kind of species that inhabit those regions are not identical, despite the seemingly identical environment. And often we can infer why this happened by examining the geography of the location. So the figure here, uh, it depicts a so-called Wallace's line. And Wallace's line, it's located in the Indian Oceans. It separates the islands in the Southeast Pacific. No, Southwest Pacific, right? Southwest Pacific. And so this Wallace's line is basically a trench And despite that, these islands here having virtually the same conditions, they have very different species. And the reason for that is that there was no migration going on between these islands here. Because even when the water levels are low, there are literally no bridge, no dryland bridge that forms among these islands, unlike like all the other islands here or here. So the result is basically a unique variety of organisms on each side of the line. So, apart from the paleontology, biogeography, which is a study of species distribution, can give clues about evolutionary events. So the clue of the Wallace's line, basically, it gives the clues that evolutionary events here were separate from the evolutionary events here. So these two groups of species, species in red and species in blue, have evolved independently. So, and despite having similar environmental conditions, they evolved like in different ways because the interaction between these species is completely different from the interactions between species in red. Okay. A little bit about Alfred Wallace that I wanted to mention. He was a British naturalist like Darwin, and like Charles Darwin, he came to this idea of natural selection completely independently and at the same time as Darwin, which shows that, which goes to show that natural selection idea is kind of intuitive once you know enough about the world. And so two people came up with it at the same time in, well, in Britain. So this British naturalist Wallace, he's the one who noticed the unique assemblages of birds and mammals in here and in here, and found that these islands are in fact separated by the deep trench, which prevents the species migration between the islands and interactions. So once we look at the fossils and at the live species as well, we look at anatomical relationships and these between various species, because these anatomical relationships can reveal common descent. So in this case, we see that in the figure here, some bones have the same colors in human, in a bat, in this bird here, not sure what type it is, in a lion and in a seal. So this group here are all mammals. The bird is not a mammal, however, we have a common ancestor with the bird, which basically um, uh, the evidence for which is some structure in the bones between us and birds and other mammals. So the structures that are inherited from a common ancestors are called homologous structures. So the bones in our upper extremity have homologous structures with those of a bat, with those of a bird, with those of a lion, and with those of a seal. So like the structure of our upper extremities is basically the same. So the structures are considered to be homologous because we have a common ancestor. We inherited that structure from a common ancestor. However, sometimes these structures can become vestigial or not needed anymore. So a vestigial structure is homologous to a functional structure in another species. An example of a vestigial structure is the hind limbs like in a whale and in some species of snakes. So some whale ancestor used to have like this pronounced hind limbs. However, the modern whale has a vestigial hind limb, meaning over time the whales evolved to have like a less pronounced hind limb because they didn't need it. And same with snakes. Actually, snakes some species of snakes have vestigial hind limbs, like the I'm gonna quote unquote proto snakes, or the species that kind of from which the snakes evolved from, they used to have hind limbs. Just over time and over evolutionary time scale, these limbs became vestigials or disappeared altogether because the snakes kind of survived best without them. Or the species of the snakes that survived tended not to have the hind limbs. Uh, Nicholas asks, is appendix a vestigial structure? Yep, it sure is. And wisdom teeth? Well, wisdom teeth are kind of there. They're not like atrophied or anything. Although there
They're
was a
becoming.
paper, there was a paper not long ago that said uh, that more and more people are born without the uh, wisdom teeth, or they're becoming very, very small. So another example of the vestigial structure is eyes in a mole. So in this picture, you see a mole, and moles are blind. They kind of don't use their eyes, so but they still have these vestigial structures that, you know, for the eyes, because the ancestor of the moles did have eyes, and moles are mammals, so they're and we are mammals, so we have a common ancestor with moles, but we have eyes, and the moles are blind. So, but they do have eye sockets. So these eye sockets are vestigial eyes. All right. Sometimes organisms appear similar on the outside. However, they do not really have a common ancestor. And if the two organisms have similar structures but do not have an ancestor in common, then these structures are considered analogous. So analogous structures evolved independently. Some An example are this pair here. So this is a salamander living in a cave, and this is a crayfish living in a cave. Both of them, uh, have discolorations, or they lack color on their on their bodies, but they do not evolve from the same common ancestor. So like colorless body is considered to be an analogous structure. So also they do not have eyes, and lack of eyes is considered to be analogous structure. Non-functional eyes are considered to be analogous structures rather than homologous structures, because did not evolve from the same, they did not have a same common ancestor. Another example that is maybe more relatable, a shark and a dolphin have very similar body shapes and very similar like fin structure, but a shark is a fish and a dolphin is a mammal. Like they did not have the same common ancestor. So the body structure of a dolphin and shark is considered to be analogous structure. And this is like another example that is given in the book. One is a cactus, and another one is a euphorbia plant. They are not evolutionarily, like genetically, they're not similar, but they do have the same structure, which is considered analogous. Like they have this succulent type of structure. So the analogous structures, they result from so-called convergent evolution. So if the conditions are the same, if the conditions in which two organisms live in are the same, eventually these organisms are likely to develop the traits that are beneficial for surviving in that environment. So like both cactus and euphorbia live in deserts. So, and this structure that they have, this structure that allows to store water and prevents evaporation of water from the inside of the plant, it is beneficial for surviving in the desert. So, well, it's actually quite vice versa. The cactus and euphorbia have this structure because um, the individuals that developed this structure survived. And over time, only the individuals that have this kind of structure will be visible because they survived. And same goes for salamander and crayfish, and same goes for dolphin and shark example. These analogous structures are the result of the convergent evolution. And during convergent evolution, species develop traits that facilitate their survival. Okay? And these traits can be very similar between very evolutionary different groups. All right, and it's 10:20, so I'll pick it up. I still have a bit to cover here, and I'll pick it up on Friday. Any last minute questions? Lauren asks, did we get to everything for the participation assignment today? Also, are we allowed to go over with Ansley which question we got wrong on the exam to learn where we're going wrong? Lauren, so I think we covered everything for the participation assignment. I'm going to check, but if not, I like some other time that I made this that that this happened. I'll just give everybody the point, the credit um, for the material that we did not cover today. And of course, you're welcome to schedule an appointment with Ansley to go over what you got wrong on the exam. Uh, Christine asks, do you have any comments from the exam? Uh, yeah, you did very well. The average score for the exam is 84%, which is great. So you guys did fantastic. Good job. Uh, any other questions? All right. Well, I'll